Okay, we'll have a little bit different schedule tomorrow since we have an exam Monday. We need to do some pre-lab stuff, which we would normally do on Monday, so that will be tomorrow. In preparation for that, there is a section in the next lab called the Employee Resource Manual. That is something you should read through, and we'll talk about some of that stuff tomorrow. But that won't take the full period, and so there will be time for some exam review. So very specifically, if you have questions, that would be a good time to get those answered. Come in.
Is there an exam review session tonight?
Yes, yeah. Austin has his normal mentoring session tonight. 8 o'clock. Today, 8 or 9 tonight? 8. Okay. Yep. We will finish up the material that you're responsible for on the exam probably in about 5 minutes. So I'll make sure that I tell you what the breaking point is, but that will take you through that 14.2 section on transcription, and then we'll pause for just a second, and then we will continue on, and that material we cover the rest of today will be on your 4th exam. Okay, so you'll have a nice little break of a few days before you have your exam for new material. Okay, so we're in the process right now of digging into the central dogma, and the information you're going to be responsible for now, will be the transcription part of the central dogma. So our genetic information encoded into the DNA is going to be transferred into a intermediary molecule, an RNA molecule. And one of the things we want to look at a little bit is the relationship between the information in the DNA and the relationship in the RNA. So we saw this particular slide last time. This shows transcription in progress. Here's our RNA polymerase molecule, which has pried apart the 2 double strands of the DNA molecule. The strand that is being copied is called the template strand. And so RNA polymerase will assemble new nucleotides in the 5' to 3' direction. And the base pairing rules that apply are A will pair with T, or U, and G will pair with C. And so this genetic information in the template strand is being copied into a complementary RNA molecule. What about this other strand up here? Did you say, Larry, that they call this in the book the coding strand?
Coding strand.
Yeah. So this is a template strand. That's the one that's being copied. This one's called the coding strand. Why do they call that the coding strand? Well, if you look at it, what's the relationship between the sequence of nucleotides in the coding strand and the sequence of nucleotides in the messenger RNA? They're the same except for uracil replacing thymine. Okay? So by copying this template strand, we get an RNA which carries the same genetic information as the coding strand. And so that's kind of interesting. We don't really copy the coding strand to get the message. We copy its template because these are complementary. Do you want to say something?
What that means is when you sequence DNA, you can know the sequence of the amino acids that would be linked together reading the messenger RNA, because the coding strand has the same nucleotide sequence as the messenger RNA, except for that one substitution.
Yep. Remember that in eukaryotic cells, we talked about the fact that this promoter region, which precedes the information that actually encodes the polypeptide, this promoter region is where the RNA polymerase attaches. In eukaryotic cells, there is a series of nucleotides within this promoter that are very heavily A's and T's. We call that the Tata box. And these other proteins, these transcription factors, which help RNA polymerase to do its job, recognize this Tata box and therefore interact with the promoter. And the binding of these proteins to the DNA helps to then properly position the RNA polymerase so it can now move down and do its transcription. This is going to be important later on when we talk about regulation of gene expression. The fact that proteins are interacting directly with DNA and helping to determine whether transcription of that DNA occurs or doesn't occur. Remember, if it doesn't occur, then that gene isn't being expressed. So the availability of these transcription factors is one of the ways that we can turn on and turn off genes. We can regulate gene expression. Okay? So that's the ending point for the exam. Okay? So now we're on to new material. RNA molecules are being produced by this process of transcription. What are some of the types of RNA molecules that are produced as genes are transcribed? Well, one type of RNA that's being produced, and the one we've mentioned most to this point, is messenger RNA. So what information is encoded into a message? What is that message telling the cell how to do? Make a polypeptide chain. Okay? So the messenger RNA contains the information for the primary structure of a polypeptide chain. To make this polypeptide, you need this amino acid, hook to this one, hook to this one, hook to this one, hook to this one. Where was that information encoded into? In the DNA, and specifically in the coding strand of the DNA. And during transcription, that information has now been kind of transferred over to this messenger RNA molecule. Well, if we've got a messenger RNA, that information needs to be turned into a protein for gene expression to have occurred. And that part of the central dogma is translation. If we've got messenger RNA and ribosomes, do we have everything we need to make a protein? We've got instructions in the message, and we've got ribosomes. So can we make a protein? No, we need some other things, don't we? What else do we need? We need some amino acids. And later on, we'll see that these particular amino acids have to be activated or charged to be able to participate in this process of making protein. So we need activated amino acids. What else do we need? Got a message, we got some ribosomes, we got some amino acids. We need two different types of RNAs to make this all work. One of the things we need is transfer RNA. And transfer RNA basically is going to pick up these activated amino acids and carry them over to the ribosomes so that we can make the protein. So that term transfer is kind of an apt term, okay? It describes the fact that it's transferring or carrying those amino acids to the ribosomes. And then, to make the ribosomes, we need a third type of RNA. And remember, that is the ribosomal RNA. So encoded into your DNA, there not only has to be information for how to make polypeptides, genes for messenger RNA, but you also need genes for making transfer RNA and ribosomal RNA for this whole process of translation to work. Okay? So that's where that modification of the concept of a gene came in, is that we used to just say, well, one gene, one polypeptide. One gene, one set of information for polypeptides. A message, basically. But now we know we also need genes for transfer RNAs and ribosomal RNAs. So the new, most up-to-date definition of a gene is that a gene is a cistron, which is a functional RNA molecule. Now, if this had been years ago, and particularly when Dr. Morton and I were taking these courses, this is where our list of products of transcription would have ceased. At that particular point in time, we knew that there were three major types of RNA produced by transcription: messenger, transfer, and ribosomal. That list is expanding. We now know that there are another type of RNAs that are produced called snRNA. SN stands for small nuclear RNA. We'll talk about what the roles of some of these snRNAs are a little bit later on, once we finish going over kind of the overview of translation. These are going to be important, especially in regulation of gene expression. Another type of RNA that's been discovered, miRNA. Mi stands for microRNA. That's also going to play a role in regulation of gene expression. siRNA, small interfering RNA. And this isn't the complete list, okay? The list of different types of RNAs is growing. And what we're finding out is a lot of these additional RNAs play important roles in regulation of gene expression. So we'll come back and talk about those a little bit more later on. Another thing that's been quite interesting that we've been discovering recently is that we know that not all genes code for messages, that there have to be genes for ribosomal and transfer RNA. But additionally, some recent discoveries have indicated that a lot of your DNA is stuff that we call non-coding DNA. It doesn't code for a message. It doesn't code for a ribosomal RNA. It doesn't code for a tRNA. And a lot of this non-coding information is stuff called introns. We're going to talk a little bit about introns later on. So that's been an interesting and kind of exciting discovery that's happened. And then, as I mentioned, a lot of these smaller RNAs, like the microRNA and the small interfering RNA, really are going to play a very crucial role in the regulation of gene expression. And we're going to see a little video later on and talk about one particular type of regulation of gene expression, RNAi. RNAi stands for RNA interference. So these are some of the kind of things we're going to be encountering down the road in Chapter 15. Okay, so let's stick with the basics for right now. Okay, let's concentrate primarily on just the messenger RNA, the transfer RNA, the ribosomal RNA, and how the message is read by the ribosomes with the help of these tRNAs to make that polypeptide chain, which is going to do something important in the cell. It's going to fold up in a particular three-dimensional pattern, and it's going to have a particular function in the cell. Let's start out with ribosomes. We call ribosomes kind of the workbench of protein synthesis. This is where the amino acids are actually physically joined together and where peptide bonds are formed that hook those amino acids into a primary structure. Ribosomes are made up of protein and ribosomal RNA. There's approximately about a 60%, 40% ratio. Don't worry about memorizing that number. That's just there for illustrative purposes. Some of these proteins that are part of the ribosomes have enzymatic properties. And they're going to catalyze reactions associated with making protein. Interestingly, though, some of the RNAs are also catalytic. Do you remember what we call an RNA molecule that has catalytic properties? About this at the beginning of the semester. Remember the term ribozyme? So ribozymes are going to play an important role in this process of making proteins, of translation. We also did talk about, when we talked about ribosomes, the fact that they consisted of two subunits, a big subunit and a small subunit. We'll look at the structures of those in just a minute in more detail. We can't make proteins without these structures. They literally are the workbench where proteins are assembled. And one of the key things that the ribosome does is it positions these activated transfer RNAs, which are carrying amino acids, and allows them to then hook the amino acids together on the ribosome. So the ribosome has a very interesting structure. The ribosome is kind of a molecular assembly line that allows these amino acids to be joined together to make that polypeptide chain. The result of translation is a protein or a polypeptide chain whose amino acid sequence is specified by a messenger RNA, and that message was transcribed from a set of sequences in the DNA that we call the gene. So, here's a ribosome. What we're going to do right now is we're going to assume that we're looking at a eukaryotic ribosome. Remember that we can characterize structures like ribosomes by a value called the S value. Does anyone remember what the S value is of a eukaryotic cytoplasmic ribosome? It has an S value of 80. Remember what the prokaryotic ribosome S values were? They were 70? Okay. So it's this difference in prokaryotic and eukaryotic ribosomes that some antibiotics exploit to selectively kill the bacteria. So this whole thing would be an 80S ribosome. Okay? It consists of a large subunit and a small subunit. The large subunit is a 60S subunit, and the small subunit is 40S. And no, my math isn't bad. These do not add up to 80. Okay? I'll typically just refer to these as the large and the small subunit as opposed to 60S and 40S. So, what are these things? Okay? These molecular machines, if you like, are a bunch of ribosomal RNAs and proteins all glommed together to make a functional unit that's going to translate the message to make a protein. What does it contain? Well, there's some important binding sites on these ribosomes. And one of the really important binding sites down here is on the small subunit, and this is where the message attaches. Okay? So this thing is going to have to physically bind to this messenger RNA and then kind of move down the message like trains going down tracks and read the code words in the message. And every time it encounters the code word, it's going to have to go AUG. That means methionine. That's what the genetic code is. UUU. That means, I think it's phenylalanine. Okay? So somehow this ribosome has to interpret what those code words are and then bring the correct amino acid into position that corresponds to that code word. So you got to have a binding site to get the message attached to this machine. There are three other important binding sites on the large subunit. And they are designated the A site, the P site, and then the E site. So if you want to think of ape, that's a good way to remember the designation for these three particular binding sites. Two of them, the A and the P, will bind transfer RNA that has been activated, that carries a particular amino acid. The third site, the E site, is kind of the exit site. And that's where they leave as the ribosome moves down the message. We'll show you what that looks like in more detail in a minute. So at any one given time, a ribosome can bind at the same time 2 transfer RNA molecules, but no more. Here's kind of a molecular model of what a ribosome looks like. Again, in blue, we've got this large subunit. Down here, we've got the small subunit. You can see that the small subunit has bound to a message and is in the process of reading that message from the 5' end, moving toward the 3' end. So this illustration is kind of in mid-sequence. It's already synthesized a fair number of amino acids by reading this particular part of the genetic code in the message, but it still hasn't read this part right here. So here's our P site and our A site, and these little squiggly things down here represent transfer RNAs that are bound to those 2 sites. Here's a transfer RNA that has been released from the message and is in the exit site on its way to then just being released from the ribosome entirely. And then here's our growing polypeptide chain right here. So this particular amino acid, this one, this one, this one, have been made by reading this genetic code in the message. It's a pretty remarkable piece of machinery. I'm going to skip this one. I don't think this one's that important. Okay, the other player in the process is tRNA. This transfer RNA is the molecule that's going to grab amino acids, and when it hooks up with a particular amino acid, it will then bring that amino acid to the message and then allow those amino acids to be strung together to make a polypeptide chain. What's a tRNA? Well, a tRNA is a single-stranded RNA molecule. That's what we mean by a single polynucleotide chain. So it's made up of a bunch of A's and U's and G's and C's. Okay? But this particular molecule is folded into a complicated 3-dimensional shape. They typically are about 75 to 80 nucleotides in length, so they're pretty small RNA molecules. Messages can be hundreds or thousands of nucleotides in length. tRNA is pretty small. What do? tRNAs bind a particular amino acid. So there's going to be some specificity between a particular tRNA molecule and a particular amino acid. What ensures that specificity? Well, there's an enzyme involved. This amino acyl tRNA synthetase. And what this enzyme's doing is it's grabbing a particular tRNA, grabbing a particular amino acid, and sticking them together. And that activates the amino acid. We'll see how that happens in a second. The tRNA then has to be able to recognize particular codons in the message. So it's going to have to, a particular tRNA is going to have to attach to a specific codon. There's a region on the tRNA called the anticodon. So if we've got a message, let's just say this is a message right here, and this little hook is going to represent a tRNA, which is going to carry some particular amino acid. We'll just call that amino acid 1. Which tRNA will attach to this particular codon in the message? The one that is complementary to the codon. This sequence of 3 bases at the end of the tRNA is called the anticodon. So by anti-codon-codon pairing, a particular tRNA positions itself across from a particular codon, and that then positions a particular amino acid into place. If another tRNA comes in here and binds to the next codon, what's its going to be? Let's say this has amino acid 3. Then this is the codon, and so this anti-codon is going to have to be that. So we always have the complementary base pairing that's occurring. So in some respects, these transfer RNA molecules are kind of adapter molecules. They adapt the language of nucleotides, genes, into the language of polypeptides. So they're kind of the molecule that, is the intermediate between instructions and polypeptide chains. How many different tRNAs are there? Well, we would kind of expect, logically, that there would be 61 different tRNAs. Why 61? Where's that number 61 come from? Remember our genetic coding scheme? How many different possible combinations of 4 nucleotides taken 3 at a time were there? 64. But remember, 3 of those don't code for amino acids. What were those? Stop. Okay, so we're down to 61. So we would think that 61 different anticodons corresponding to 61 different tRNAs would be needed to recognize the 61 different codons, right? Well, we'll come back to this later on, but in reality, there are only 45 different tRNAs. There's this interesting phenomenon going on called wobble, and we'll talk about wobble later on. But there are more different tRNAs than there are amino acids. Why? Because the genetic code is redundant. Okay, so here's what a tRNA looks like, conceptually. Here's the 5' end. Here's the 3' end. It is a bunch of G's, C's, A's, and U's joined together by phosphodiester bonds to make a single strand of RNA, but this single strand of RNA folds up into a complicated 3-dimensional structure. Why? Because of hydrogen bonding between nucleotides in the molecule. Isn't this U or C going to be attracted to this G? Aren't they complementary? Yeah. Isn't this U going to be attracted to this A? So because of this hydrogen bonding within the RNA molecule, this folds up into this complex 3-dimensional structure. And this 3-dimensional structure is well adapted for the role that tRNA has to play. These are all representations of a tRNA molecule, as is this. Okay? What are the key features in all of these different representations? At this 3' end down here, there is a particular amino acid attached to that particular tRNA. So that's one important feature. A specific amino acid attached to a specific tRNA. The other key feature is this region down here, the anticodon. And that is a series of 3 nucleotides which will then interact with the code word in the message. All the rest of this stuff is kind of just window dressing to properly organize the molecule so it can do its job. So, how do we get a particular tRNA hooked up with a particular amino acid? Well, we need an enzyme. This amino ACL tRNA synthetase. And there are 20 different enzymes. What's significant about these 20 enzymes? What's significant about the number 20 in this context? number of amino acids, okay? So there's a different enzyme for each of the 20 amino acids. This charging process of hooking up an amino acid to a transfer RNA requires energy. And the energy that it requires is ATP. One of the things I'm going to be doing as we go through this process of translation is give you some insight into the amount of energy needed for translation to occur. Making proteins is expensive, energy-wise. And that's why it's important for cells to not make proteins when they don't need to be able to turn off genes when they're not needed. So here's what the enzyme conceptually looks like. Here's one of the 20 amino ACL tRNA synthetases. And it's going to have a binding site for one of the 20 amino acids. So there's a complementarity between a particular enzyme and a particular amino acid. ATP is going to bind. And the ATP, when it's hydrolyzed, is going to donate a phosphate group to this amino acid. So the amino acid is going to become phosphorylated. Then this enzyme will recognize one tRNA molecule. That will bind to the second binding site. And then this phosphorylated amino acid will form a bond that joins it to the transfer RNA. Once that happens, the enzyme releases this activated amino acid now. It's hooked up to a specific tRNA. And this particular tRNA will have a particular codon. Okay? Sorry, anticodon. So this anticodon information down here basically determines what amino acid is hooked to what transfer RNA. So we've got all of these charged, activated tRNAs now floating around in the cytoplasm. And if there is a message that has been transcribed, and that has left the nucleus and hooked up to a ribosome, then these charged tRNAs can interact with that message, bring the proper amino acid into position, and then peptide bonds will be formed to join those amino acids together to form the polypeptide chain. It's really important that these enzymes work properly and that they join together the proper tRNA with the proper amino acid. If they don't do that, then the genetic code is going to get all screwed up. And whatever information is encoded into the DNA will not get turned into the correct polypeptide chain. So sometimes people call this tRNA amino acid complex the second genetic code. Okay. Third player, messenger RNA. Messenger RNA contains the information for the primary sequence of a polypeptide chain. What is a message? It's a bunch of codons. What's a codon? A series of 3 nucleotides that specifies a particular amino acid. The message has to be able to bind to the ribosome. We said that that binding site is on the small ribosomal subunit. But the message also has to be able to bind to specific tRNAs in such a way that the codon-anticodon pairing is correct. So we can't violate this codon-anticodon pairing if the process is going to occur properly. So here's what it kind of looks like in progress of making a protein. So here's our message. In brown is our ribosome. In green right here are transfer RNAs. In light blue are amino acids. And these would represent transfer RNAs that have been activated, attached to a specific amino acid. Ribosomes have binding sites, the A site, the P site, and the exit site, the E site, as well as a binding site for the message. In this particular illustration, this particular portion of the message has already been read or interpreted and turned into a sequence of amino acids joined together by peptide bonds. This part of the message right here is in the process of being read or has not yet been read. And I don't know why they've got the arrow pointing this direction. I think that is indicating that the ribosome is moving this way, but direction of synthesis occurs in the 5' to 3' direction.